Hello lovelies, in this video we're going to be going through your OCR-A AS Biology paper 2, the paper that we've written for 2024. So there are lots and lots of exam skills as well as the knowledge that we want you to go through in this. It is really, really important that you can work through this quickly and effectively.
The image shows a blood smear viewed under a light microscope. Give the letter that represents a lymphocyte. Explain your answer. So hopefully we know that's a type of white blood cell. The majority of the disc shapes in this image are going to be red blood cells. X is a phagocyte or a neutrophil, depending which one you want to use, but it's basically, we know that it's one of those because it's got a multi-lobed nucleus. We can see it's kind of a string of beads almost like, so it allows it to stretch to engulf a pathogen. And then we've got Z must be the lymphocyte because it has a large circular nucleus. So our answer is Z. A biologist is studying the concentration of red blood cells in a 10 milliliter sample to assess the health of an individual. To do this, they used a hematocytometer grid, viewed under a microscope. The grid is divided into multiple squares, each with a known volume. The volume of one large grid square on the hematocytometer is 0.1 millimeters cubed. The biologist dilutes the original blood sample with a saline solution in a 1:20 ratio. Using the hematocytometer, they count the number of red blood cells in 5 of the large grid squares. The average number of blood cells counted in 5 squares was 8.18. So remember, one large grid square is 0.1 millimeters cubed. Explain why the biologist had to dilute the blood sample. So this is a similar question that you might get if you're sort of talking about diluting a bacterial broth to count the number of bacteria in it as well. Anything like this, we've got lots of floating around cells or substances in there and we want to try and count them. Diluting them makes it easier to see or easier to count them because there's need to not be too many of them in the grid. If there's loads, it's all crowded, you can't tell one individual bacteria or one individual red blood cell from another one. How can you actually see them to count? So by diluting it, we make it easier to count them, easier to see. Calculate the total number of red blood cells in the sample. Give your answer in standard form to 3 significant figures. Okay, so we need a total number in the sample. The sample is 10 milliliters, remember? And we've managed to do a sample. We've got an average of 8.18, which is 8.18 on average in 0.1 millimeters cubed of the diluted solution. And we've got a dilution factor of 1 in 20. So we need to multiply the average by the dilution factor, or multiply the average by 0.1 millimeters cubed, then multiply that by the dilution factor, and then multiply that up to get the total volume of the sample. So 8.18 times 0.1 millimeters cubed means we have 81.8 red blood cells per millimeters cubed. Then we're multiplying by the dilution factor, which is 20. That gives us 1,636 cells per millimeters cubed of sample. And there's 1,000 millimeters cubed in a milliliter. So 1,636 times 10,000 millimeters cubed gives us the final answer, which is 16,360,000. We need to put that in standard form to 3 significant figures. So we're going to do 1.64 because we can round up that 3 because of the 6. And then it's times 10 to the 7. If you're always unsure about the jumps, I do the little jumpy thing to make sure I'm happy with the decimal point. So where the decimal point is in your standard form to the end of the number is 7 jumps. That means it's 10 to the 7. Blood smears can be used to diagnose blood disorders. One inherited blood disorder known as ovalocytosis results in 20 to 50% of erythrocytes being elliptical in shape, similar to some bacteria. So if we think about this, and sometimes I like to do this in exams, it's literally sometimes the easiest thing to do is a little sketch of what is being described to you. So instead of having our like biconcave disc-shaped red blood cell, we've got sort of long tube or pill-shaped red blood cells that have been stretched into this ellipsis. So it wants us to explain how this shape change could cause reduced oxygen transport. So think about adaptations of red blood cells and their shape for oxygen diffusion, because we know that obviously this biconcave shaped disc is there as a really important part of the way that red blood cells work. So this biconcave shape normally gives them this large surface area to help maximize the diffusion of oxygen into and out of the red blood cells when they are picking it up or dropping it off wherever it needs to be. So if we've lost that, then we've lost that biconcave shape, we've lost that large surface area, we might have reduced it, therefore there's going to be less oxygen diffusion. So loss of biconcave shape could reduce the surface area to volume ratio, meaning less oxygen diffuses into the cell. Treatment can include folic acid supplements because folic acid, or vitamin B9, is essential for red blood cell production in the bone marrow. Suggest 2 symptoms someone may have if they have a folic acid deficiency. So what happens if they don't have enough folic acid in the body and it wants symptoms? So that's like feelings or physical changes in the body that you'd expect to see as a result of this deficiency. So it tells us that we need folic acid for red blood cell production. So if you don't have enough folic acid, then potentially you're not going to have enough red blood cells. This means we're going to reduce the oxygen in blood, or the oxygen transport around the body, because obviously it's linked to that first part of the question. So what symptoms could you have if you don't have as much oxygen in your blood or as much oxygen travelling to all of your tissues? Could be something as simple as being tired or fatigued because you're not getting enough oxygen for respiration for all of your cells. Could feel breathless, so your body tries to increase your breathing rate to try and get more oxygen into your blood because there's not enough. Or you could feel dizzy or faint, again, because not enough oxygen is getting to your brain. So there's a lot of similar answers to this. As long as, if there's anything that's about gas exchange, reducing oxygen concentration in the blood, reducing oxygen transport in the blood, anything to do with that, the symptoms will always be the same, because it's always about reduction in oxygen getting to certain places, causing either tiredness or dizziness or breathlessness. So I've gone for feeling dizzy and feeling tired or fatigued. You've got slightly different wordings and things in the mark scheme as well, so just making sure that you pick 2 of these that you're happy to kind of explain and that you understand, and that can be kind of your go-to for some of the questions that are like this. The diagram represents the structure of an antibody. Give the name of the part of the antibody labelled A and B. So A is the variable region because it's where that binding site is, and you can see there's obviously a difference in shape at that end of the antibody. You've got that kind of dark outline to show that that is the variable region. And B is a disulfide bond. It's holding 2 of the light chains together. So we should hopefully know that the polypeptide chains in an antibody are being held together by disulfide bonds. The heavy chains are obviously the longer ones, and that would also be the constant region as well if it was pointing down there. And the little short ones are the light chains as well. So this is just a way of remembering all of the labelling, all the bits, so we're happy that what we've got is right. The hinge region is obviously kind of the squiggly lines as well, so they can bend and twist. So sometimes with a diagram it helps to label everything you know, so then you're confident that what you have written down is correct. So we've got the variable region and disulfide bonds or bridges. Give evidence from the diagram to explain why antibodies have a quaternary structure. This is really just the definition of quaternary structure, so that's more than one polypeptide chain joined together. And so what can we see from the evidence from the diagram? Well, we've got polypeptide chains labelled. There's 4 individual polypeptide chains that have been joined together by bonds, and that's what's giving us the evidence that it's definitely a quaternary structure protein. Radioimmunotherapy, or RIT, involves producing monoclonal antibodies that have a small amount of radioactive material attached. These special antibodies are designed to find and attack tumour cells directly, delivering radiation therapy only to the areas that need it, rather than treating the entire body. Explain why antibodies used in RIT will be identical. It's that these are identical antibodies because they all came from a cloned B cell or plasma cell. So when the B cell or plasma cell goes through clonal expansion or it multiplies by mitosis, that's a form of cloning, all of those B cells, plasma cells will then be identical and they will form these identical antibodies. This makes sense because the antibodies will all need to be identical in their variable region in order to bind to the same antigen. Explain why these RIT antibodies only attack tumour cells. So it's explain questions, so we need reasons why. Why will it only attack tumour cells? So in other words, why don't they bind to every cell or any other cell other than tumour cells? There's obviously a reason why we got to label an antibody at the start of this question. It made us point out the variable region, and we should hopefully know that the shape of the variable region is going to be specific tertiary structure that's only going to be complementary to antigens on the tumour cells. So it's only going to bind there and it's not going to be able to bind to others because they won't have that specific antigen, so they won't have the right shape. And this is really just about shapes fitting together and being able to explain how antibodies work. The variable region of the antibodies has a specific tertiary structure that is only complementary to antigens on tumour cells. Now, it referred to tumour cells back in the first part of the question, so that's what we can say here. But if you said cancer cells, that's also fine. This means the antibodies can only bind to tumour cells to form antibody-antigen complexes. So the word only is doing a lot of work here because only attacking tumour cells is what is in the question, and we need to say why it's only then going to be complementary to antigens on tumour cells, which means they can only bind to tumour cells and form their antibody-antigen complexes. And you just need to make sure that if there's anything where it's like, why is this enzyme, or why is whatever it is, a shape fitting together specific and only does this, we should be replicating that idea of only in our answer to make sure we understand that it's kind of unique. Antibodies can defend the body against pathogens in a number of ways. Describe the process of agglutination. So it's a describe question, so we need steps with detail. Hopefully we've heard of this word agglutination, and it's where we've got a specific type of antibody called agglutinin, and that will join multiple pathogens together because one antibody joins to two, at least, pathogens, and therefore it can join them together and form a clump, which means it's more easily consumed by a phagocyte because it can go along and scoop up multiple at once. So agglutinins, or if you didn't remember that, you could just say the antibodies, bind to more than one pathogen, forming a clump. This allows multiple pathogens to be engulfed by one phagocyte. Again, you've got slight variations for marks in the mark scheme, so as long as you've gone along the lines that you've got there with the 2 points, 2 separate points, then you can get the 2 marks. Describe how a phagocyte identifies and destroys a pathogen it encounters in the blood. So it's a describe how, so we need to give the steps in the process and detail, so we're thinking about our key words here as well when we're doing this. So how it identifies, that's how it detects it. How is it going to know that it's a pathogen? And then how does it destroy it? So this is the process of phagocytosis. So it's really mostly going to be just talking about and describing how phagocytosis occurs. It's going to be able to identify a pathogen because it's going to recognize the foreign antigen on the external membrane of the pathogen. And then once that has been detected, it's going to trigger phagocytosis, where it's going to engulf the pathogen. Then it's going to be inside the cell, inside a phagosome. Then we have a lysosome, which is going to fuse to that phagosome. You can even refer to it as a phagolysosome at that point. And the lysosome is going to contain lysozymes, which are enzymes which will then digest or hydrolyze the pathogen in order to destroy it. So this is kind of literally 4 or 5 key words, and that's all I need as a sort of memory trigger reminder to make sure I'm going to get all of this really key language into this question and do it in the right order. So the phagocyte detects the pathogen by recognizing the antigens on the cell membrane surface as foreign or non-self. The phagocyte will then engulf the pathogen and surround it in a special vesicle called a phagosome. A lysosome will then fuse with the phagosome, where lysozymes are released inside to digest the pathogen. Okay, so we've got plenty in there without having to write loads, but every single thing I've said, all the keywords are correct. I've really been careful about spelling them. So like lysosome versus lysozyme, they mean two very different things because one is a specialized organelle inside the cell and one are a type of enzyme. So be careful and just make sure you've got all of that detail and all of those keywords. Students wanted to investigate the rate of uptake of water from a plant shoot in different conditions. A pedometer is a piece of apparatus that can measure the uptake of water by a plant shoot. The student assembled a pedometer similar to the one shown in the image below. First question says, explain why the students cut the shoot underwater. So it needs us to explain the reasons why they did this. So hopefully we know how to set up and prep a pedometer, but the reasons behind why you would cut the chute underwater is so that you don't introduce any air bubbles into the xylem vessels, and you need that continuous column of water to be kind of all the way through the apparatus, all the way through and into the chute. So to prevent air bubbles entering the xylem, so a continuous column of water is maintained. Technically either of those will still get either mark, you don't have to say both. Explain how the students could have used the apparatus to make repeated measurements and how this would have improved their data. So in order to take repeats, you would have to be able to reset the apparatus back to the start so that you could measure again for a set amount of time. So as well as explaining how they could do that, we also need to explain why doing repeats is a good thing. So to reset the apparatus, you can use the reservoir, open the tap, let water into the system from that point, which will push the air bubble back to the start of the scale. So then you can start again, because otherwise obviously your air bubble would continue past the scale and you wouldn't be able to get a measurement. So the reason why we do repeats is because we can get more reliable data or more reliable results. If you can repeat something multiple times and show that you're getting the same pattern or the same or similar results each time, and you can identify any anomalies and remove them, then your data is going to be more reliable, because it means if someone else did the same thing, they would also see the same results. So we're going to say we're going to open the tap to push the air bubble back to the start of the scale. You can add that. And then repeats make data or results more reliable. The results are shown in the table below. So we've got the time in minutes, we've got the distance moved by the air bubble in millimetres. It says the diameter of the capillary tube was 1.4 millimetres. Use this information to calculate the rate of water uptake in millimetres cubed per minute to complete the table. So millimetres cubed is obviously volume, so we need to use pi r squared and then times that by the distance the bubble moved each time to show the volume of water that was taken up. And then we're going to divide that volume by the time it took. So pi times 0.7 squared times the distance divided by time. First row is 0, so we leave that as 0. Then for the second row, we're going to do pi times 0.7 squared times 2.10 divided by 4, which gives us 0.8081. Now we need to make sure that all of our values in the table are to 2 decimal places, because that is what the rest of the values in the table are. So we just match, you always match the decimal places in the table. So I'm going to continue doing the rest of the table. So pi times 0.7 squared times 5 this time divided by 8 gives us 0.9621. That can be rounded to just 0.96. So as you can see, I've done exactly the same for the others. So I've ended up with 1.01 for 12, 1.07 for 16, because we have to round up that 6, and then 1.09 for 20 minutes. Using the data in the table, plot a graph showing the rate of water uptake. So when we're plotting graphs and when we're drawing graphs like this in the exam, just making sure that we're going to do all the things we make sure we're going to need to do to get marks. So we need to do suitable scales, we need to do our axis titles, which we're going to use exactly the same language as in the headings of the table. We need to plot our points, and then we need to draw a line of best fit. So I've just written myself a little list to remind myself and make sure I do all of those things before I finish this question. Okay, so I've put the time in on the x-axis. There is a slightly different scale in the mark scheme where they've gone in 5s. Either one is fine and should be fairly easy to do. And then on the y-axis, obviously we need to do the rate of water uptake because it wants the graph of rate. So we know we've got to do the numbers we're dealing with here. But it's fairly simple. We can go up in 0.2s and we should be able to fairly easily do that. If you did a slightly different scale, that's fine, as long as ultimately the kind of shape of your graph that we're going to get at the end looks fairly similar to the one in the mark scheme, and the scale that you've chosen is linear, is fine. Okay, so plotting my points. Each one of these little squares is going to be worth like 0.04. So for 0.81, we can go between 0.8 and the first square. So the only one of these points that needs to be directly on a line instead of halfway between two lines is going to be 0.96, because that is obviously 0.04 below the 1.0 line. And then we've obviously got between, just above the 1 will give us 1.01, and then above the 1.04 is going to give us 1.07, and then above the 1.08 is going to give us the 1.09. So you can see that we've got kind of an increase, and then it levels off towards a flat line. So our last thing to do is draw a line of best fit. It's not always going to be perfect. In this case, we can have this nice kind of smooth curve going down and meeting 0 at the end, and we should be majority hitting every single point as we go, because it should be easy and smooth to do so. The student repeated the experiment with a fan placed next to the plant shoot so that air was blowing over it. Sketch a line onto the graph to show the results you would expect with the fan. Label this line with fan. Okay, so we need to think about what we'd expect to see, what would our graph look like if we repeated the experiment with a fan. So there'd be increased air movement around the shoot, which would increase the transpiration rate, so the evaporation of water from the leaf. So we'd expect that to then show as an increased rate of uptake of water. So in terms of what that would look like, we need a steeper initial rate. So we need a line going straight up, and then it would need to be higher than the line we've already drawn. But we should show the same pattern. So there we go, I've drawn it, I've made sure it's above the original line the whole time, and it's following a similar pattern, and I've labelled it with fan. Now it wants us to explain the differences we'd expect to see in the results with the fan. So we're basically given the reason why we've drawn the line the way we have, and we've already kind of explained this when we've said about the fact that it increases the air movement, so we're increasing the rate of uptake because we're increasing the transpiration rate. The reason why it's increasing the transpiration rate is because it's decreasing the humidity, or you can talk about maintaining the concentration difference between the water vapour that's then evaporating. Water uptake rate would increase because the fan increases air movement around the leaves, reducing humidity and therefore increasing the rate of transpiration. And what we've said is that would obviously increase the rate of water uptake. So you've got different wording here, you've got some different wordings in the mark scheme as well, but as long as you've got this idea that you're increasing the air movement and what that does to cause a greater loss of water at the leaves, and therefore that means that the plant would need to take up more water. Human activity is impacting biodiversity. Peat bogs are unique wetland habitats for many species. Peat bogs are being drained for human use, including grazing of livestock. Peat can also be cut into blocks and extracted for use as seen in the image below. Explain what is meant by the term biodiversity. Explain what is meant by the term questions are just define questions. So biodiversity is described as the variety of different species living in an area. And you've got various options for talking about area, habitat, ecosystem. As long as you're talking about the fact that you're talking about the number of different or the variety of different species, that's what we mean by biodiversity. Explain how human activity has reduced biodiversity in the peat bogs. So it gives us some clues in the question. It's talked about the fact that they've drained it, so they've removed the kind of wetland element, they've drained the water, they've got animals eating the plants through grazing, they're also removing the peat itself. So this question has got some clues in the question to help you, and also we may remember about peat from GCSE, but it's about making sure that you're explaining why those things are reducing biodiversity specifically. So reducing biodiversity means we're reducing the number of different species or organisms in that peatland area. So we've got always the same answer. If something's talking about why is biodiversity being reduced, we've got the same few things we can talk about. There's fewer habitats, there's fewer niches, there's fewer food sources. And you have to say food sources, not just there's less food, because it's not about the amount of food of a single species overall. It's about that there's fewer different options of species that they can use for food, and that reduces therefore the number of organisms that eat that food or rely on that food. So there's fewer food sources, and we can link that to the explanation of because plants are being removed because of the grazing animals. We can talk about the fact that there's fewer habitat, or the habitat has changed because they've reduced or got rid of the wetland element and they've dried it out. So that means that species are adapted to live there can no longer live there. There's lots of ways we can explain this, and you've got lots of different options in the mark scheme. I'm going to go with fewer plant species due to grazing or extraction. So we've literally removed plant species in that way. And that also means there's fewer food sources. The draining of the wetland also reduces the unique habitat, so specialised species can no longer survive. Okay, so I've actually probably done more than 3 marks worth potentially, because I've got fewer plant species, fewer food sources, and the changing of the habitat, but that is 3 marks. And you can obviously talk about more things, but this is the kind of standard answer that we would normally try and apply to a biodiversity reduction question. Give 2 reasons why biodiversity should be maintained. So why should we maintain biodiversity? It also goes for conserving it, protecting it. Why should we try to keep high biodiversity? Why is high biodiversity important? So there's a few reasons we should maintain biodiversity. The first one mainly is the moral or ethical reason that we shouldn't just let species go extinct as a result of human activity. That's not okay. We need to safeguard those species for future generations. Those species in high biodiverse areas could provide resources for us, medicines for the future that we haven't yet discovered, other resources that we use plants and other organisms for, and also potentially like we make money off of them through trade or ecotourism, so there's a financial incentive as well there. There's lots of reasons in this mark scheme and kind of the way you can word it, but I'm going to go with sort of 2 about the kind of moral, ethical, and the resources. So biodiverse habitats could be a source of future medicines for humans. We have a moral duty to prevent species from going extinct. A sustainable alternative to fossil fuels is biofuel or bioethanol. Cellulose is a carbohydrate found in the cell walls of plants. Cellulase is an enzyme produced by bacteria, fungi, and some protists that can digest cellulose. Cellulase is used in the process of making bioethanol to obtain sugars for fermentation. Suggest the type of organism that produces cellulase. So this is a suggest question, so we are using our own knowledge and some knowledge given to you by the question, but it's basically asking kind of what type of organisms are these bacteria, fungi and protists that will be able to produce this cellulase enzyme and therefore be able to digest plant material. So this is kind of the clue here. It's like we've given you some microorganisms and ask what you think they might be able to do or what type of organism they might be if they can digest plant material. So I would suggest my first thought would go to plant pathogens, because then they can break through cell walls in order to get inside the plant, but also decomposers, because they break down dead organic material from plants in order to recycle the nutrients, and so they're probably going to have cellulase as an enzyme. So either of those answers, because it's a suggested question, there isn't always one specific right answer, will get you the mark here. Cellulase is used in the process of making bioethanol to obtain sugars for fermentation. A student recreated this process in the lab by grinding a sample of shredded paper with some water to create the pulp. They set up 3 test tubes as follows. Tube A contained the paper pulp and water, tube B contained paper pulp and 5% cellulase solution, and tube C contained paper pulp and 5% rubbing alcohol. So we know from the start of the question that cellulase digests cellulose, which is going to be in the cell walls of the plant material that was used to make the paper. And then we've got 5% rubbing alcohol. It's a solvent. It could disrupt hydrogen bonds between the chains of cellulose, or potentially interfere with ionic bonding, but I think we don't necessarily need to know that, but it's something I would write down to maybe think of later in case you get asked the question. The tubes were left overnight. Describe how the student could test the solutions to see if sugars had been released from the paper pulp. So it's a describe how, but it's based around a practical, so we kind of want method steps, what are they going to do in detail. It says they're going to test the solutions, so what was in each tube, and it wants us to tell us about how could they test it for sugars. So hopefully we know this should be a Benedict's test, and because it's detailed, we need to say what they would do. So how would they actually test it? So we need to add the Benedict's, and then we need to heat it. So specifically, we either need to say boil, or we need to say heat to above kind of 60, 70 degrees in order to make sure that that reagent's actually going to work. Add Benedict's reagent to a sample of the solution and heat it in a water bath above 60 degrees. Now it wants to suggest and explain the result you would expect to see in tube B after testing for sugars. So suggest and explain is basically saying, what do you think you would see and why? So predict the result and explain to me why you would expect to see that. So if we've got cellulase in there, that's going to digest the cellulose, which will release beta glucose, okay, because we know that cellulose is made up of lots of beta glucose molecules joined together. So then that should be, glucose being present, change the reagent to a red-orange colour. So the Benedict's reagent would turn from blue to red because the cellulase enzyme would have hydrolyzed the cellulose to release the beta glucose. So the hydrolyzed here is a really key point. It told us that cellulase enzyme breaks down cellulose. We are adding the extra information that we understand it would be a hydrolysis reaction that would break apart the glucose molecules. You could even add, obviously, glycosidic bonds because obviously it's a carbohydrate. But this idea that we're pulling together our knowledge of the structure of cellulose, the bonds that hold cellulose together and the fact they would be hydrolyzed, the idea of how enzymes work, and our knowledge of food tests all in one 3 marker. But this is something we should be able to do with like carbohydrates, lipids, proteins, know the tests, know the structure of the individual biological molecules, how are they joined together, how are they broken apart, and that's all the information we're kind of pulling together from that topic. The diagram shows a molecule of ATP. Describe how an ATP molecule is synthesized in cells. So describe how questions are always questions that want steps in a process but with detail. So if it's asking how ATP is synthesized, that's just asking how is it made, how is it produced. So hopefully we know the basics is that it's made from ADP and PI being added together, but how does it actually then come together to form ATP? Well, it's a condensation reaction, which obviously is then going to release a molecule of water. So anything, and this can be a common question in A-level exam papers about how a molecule is made. If it's made with a condensation reaction or broken down with hydrolysis reaction, that's what it wants you to talk about. The condensation and then releasing the water molecule, or using a water molecule to break apart a bond through hydrolysis reaction. That's the level of detail we're talking about when we can talk about when we're making or breaking molecules apart. So ADP is combined with PI, or inorganic phosphate, in a condensation reaction that produces a molecule of water. Give 2 properties of ATP that make it a suitable source of energy in organisms. So 2 properties, these would be the result of the structure of ATP. So what 2 kind of features does ATP have that make it a suitable source of energy? So what's useful about either its ability to be stored, its ability to release energy, its ability to, whether or not it affects, it can move around because of its size, or how easy it is to be made or synthesized. All of these are kind of properties. So the ones we care about, and the few things we should hopefully know about ATP, are that it's easily made and easily broken down. It's one simple reaction. It can release energy in small amounts where it's needed, so we're not getting a big sort of combustion reaction where we're releasing loads of energy at once, because that would be dangerous. So they're the 2 things that I think are the main things we should know and think about ATP. There are other things on the list, so if you've got something else that's on the mark scheme list, then as long as you've got 2 of those, that's fine for G marks. So it allows the release of energy in small amounts where it's needed, don't necessarily need that part, and it can be easily synthesized and it's made in one simple reaction. You can also flip that around to say it's easily broken down as well. Compare the structure of ATP and DNA nucleotides. Okay, so structure, we need to think about molecules that make them up, bonds, that's what we're looking at, and it's a compare question. So we're looking for mostly differences, but we can also talk about the similarities because they are very similar molecules. So I've drawn a generic DNA nucleotide and then ATP as a rough sketch, just to kind of help myself think about the answer to this question. So in DNA, the base can be any of the 4 bases, but in ATP, it is adenine. That's what the A stands for. The sugar in DNA is deoxyribose. The sugar in ATP is just ribose. And then we've got 3 phosphate groups in ATP, but we've only got 1 phosphate group in DNA. So there's my 3 differences. There's my 3 marks. But obviously, they both contain phosphate groups, they both contain a sugar, and they both contain a base. So we've got the 3 similarities, and then you can sort of use those as your start point, talk about all of these 3, and making sure that we're giving the similarity that they both contain a base, for example, and then the difference. They both contain a sugar, and then the difference. They both contain phosphate groups, and then the difference. So both contain a sugar, but ATP contains ribose, and DNA nucleotides contain deoxyribose. Both contain a nitrogenous base, but in ATP it's always adenine, and DNA nucleotides, it could be any one of the 4, so A, C, G, or T. Both contain phosphate groups, but ATP has 3 and DNA nucleotides only have 1. The diagram shows the phylogenetic tree of part of the cat family Felidae. State the genus of the clouded leopard. Okay, so we've just got standard phylogenetic tree. Just need to find the clouded leopard and write down the genus, which is Neophelis, because it is the first part of the binomial name. Be careful when copying, make sure you don't copy it incorrectly, and you need to get the capital letter for the N, because otherwise you wouldn't get the mark. This is just a copying exercise, so they're going to be really strict with it, otherwise it would be too easy. A student stated that all of the leopards must be more closely related to each other than to the other big cats, as they all have leopard in their name. Explain whether the phylogenetic tree supports their conclusion. So their conclusion is all the leopards must be more closely related to each other than any other big cat. So let's first of all check that we can find all the leopards. So we've got the first one is the leopard, then we've got the snow leopard, and we've got the clouded leopard. The leopard is more closely related to the lion and the jaguar because they share a more recent common ancestor than it is to any other leopards. The snow leopard will be more closely related to the tiger because they share a more recent common ancestor than the others, than any other leopard. And then the clouded leopard is on a branch on its own, so it evolved the longest time ago, or separated the longest time ago, and it's in a different genus entirely, so it's not got Panthera as its genus, it's got Neophelis, which must mean it's significantly different enough from the others to be classified in a different genus entirely. So no, the student is wrong, and this tree doesn't support their conclusion, but we can use all these points. There is, however, obviously, that they are all related. They do share a common ancestor that was more recent than other groups, which is why they're all in this same family. And so they will have shared ancestry. But in terms of being more closely related to each other, all the leopards, than they are to other big cats, that's just not true. So we can say that first, all of the leopards share a common ancestor and they are all in the same family, but, and then we're going to give our reasons why this doesn't support the conclusion. They are not all more closely related to each other. The clouded leopard is significantly different enough to be in a different genus. And the snow leopard is more closely related to the tiger than to another leopard. So I've only used kind of 2 of my 3 main points that we're saying about the differences, but then I've also talked about the fact they do share a common ancestor, so they are related in some ways. But what I have given is enough for 3 marks. You've got the mark scheme there with the options, so if you just spoke about the differences, that's also fine. The snow leopard is quite different from the other cats. It lives in habitats that are very cold, snowy, and mountainous. Describe a physical adaptation the snow leopard might have that would help it to survive in such environments. So it's obviously about preventing heat loss or staying warm in a cold and snowy environment. There's many things that we could talk about, but we basically just need to think of something that we can relate to reducing heat loss or helping to maintain body temperature. So thick fur, fat insulation layer like other organisms that live in cold weather or in cold water like polar bears, or a small surface area to volume ratio to reduce kind of the diffusion and radiation of heat from the body. The survival of the snow leopard in the wild is under severe threat. These threats include poaching for their fur, which is sold. State the name of the international agreement that is helping to protect snow leopards. So the clues to this are the fact that it's talking about trading or selling, poaching them for their fur, which is then going to be sold. So that's about trade. So it must be CITES because that's to do with trade in endangered species. Give 2 aims of this international agreement. So what do they try and do? What do they try and help with? You've got a long list in the mark scheme because there's sort of different things you can say about what they do, and you can choose to kind of learn a couple of these that you're happy with for the future for answering questions about CITES. But the main one that they obviously do is they prevent trade in endangered species or parts of endangered species. And they also restrict then the international trade of any products from wild animals. So it protects endangered species from the illegal wildlife trade, and it restricts international trade in wildlife or wildlife products. The range of the snow leopard habitat stretches across parts of 12 different countries, suggests why the rules of the agreement may be hard to enforce. So again, there's clues in the question because it's this suggest question, so we're using our own knowledge and things to try and help us answer this, but it's telling you it's across 12 different countries. That's a massive area, and that's lots of different governments and countries coming together. It can be hard to get them to actually work together, to cooperate, to communicate, to make sure everyone's doing the same thing. Again, lots of different answers for this in the mark scheme because, again, this is a suggest question, so there isn't exactly one answer. But anything where we're thinking about trying to coordinate between lots of different countries, or we're trying to police a really large area, why does that make it hard to actually enforce these rules? So I'm going to go with can be hard to get so many different countries to communicate and work together.
Ouch! This is why in some videos I explain scratches.